Hi, my name is Anabel Chang, and I'm in the Marcus Lab, where I'm involved in a project where we are characterizing conformational fluctuations of DNA. So, local fluctuations of the sugar phosphate backbones of DNA are a form of something called DNA breathing, and these fluctuations play an important role in protein DNA assembly and enzymatic function. These fluctuations contribute towards the formation of DNA structures, which can be recognized by proteins during DNA replication, which allows proteins access to the interior bases, and it also allows sequence-independent binding. And our lab aims to characterize the main structures and their fluctuations involved in functionally relevant DNA topologies, or how the DNA is twisted and folded, and we focus the most on single-stranded, double-stranded DNA fork junctions. So to do our experiments, we have to label our DNA, and the DNA is internally labeled, which means that a fluorescent dye is inserted into the sugar phosphate backbone, as you can see in Figure 1 under Materials and Methods. And we label the DNA at specific positions relative to the fork junction with either a Cy3 dimer or monomer, like Figure 2. And a fluorescent dye, like the Cy3 that we use, gets excited by a certain light and re-emits that light as fluorescence. And in our experiments, we track that emitted fluorescence. And also under Materials and Methods, in Figure 3, you can see an experimental single-molecule spectroscopic method, which was developed in our lab to monitor the local fluctuations of Cy3-labeled DNA constructs. And we use a linearly polarized continuous wave beam to excite the sample, which is the DNA molecule, so that the emitted fluorescence contains information about either the rotational motion of a Cy3 monomer-labeled DNA, or the relative conformational changes of Cy3 dimer-labeled DNA. And by monitoring the fluorescence from the labeled DNA, it's possible for us to study local conformational fluctuations within the DNA at specific sites. So using the data from this experiment, we can make time correlation functions, or TCFs. And in these three graphs here, Figure 4, there are second-order time correlation functions, where there are two successive observations with a time difference tau in between the observations. And these 2-point TCFs tell you the average change in correlation of observations in time, or specifically here, it's telling us how far away from average the phase factor quantity is at time t compared to a time of t plus tau, or t plus the time difference. And in figures 4a, 4b, and 4c, we are measuring the same correlation function at 3 different positions in the DNA, which you can see next to each TCF. And the correlation, depending on the position, is different. And there's a loss of correlation over time, which you can see by the decay in each of the curves, which look like they also have exponential components. So with these functions, we fit exponentials to the curves that you see, and the time constants of the exponentials tells you about the timescale of the fluctuation. And these timescales are how long it takes for the system to lose its correlation, or how long it takes for the system to be in different states. And whenever it stops being correlated, that tells you that the fluctuation must be happening within that time difference, because the system is no longer in the same orientation. So this experiment ultimately tells you about the dynamics of the DNA fluctuations. And as this polarization sweep experiment is wrapping up, we're also looking towards the single photon phase tagging for single molecule Fourier transform electronic spectroscopy experiment. And in Figure 5, it shows the setup for this. And from the experiment, you get the measurement of a single molecule's absorbance spectrum, which you can see in Figure 6b. And then we can use models to fit that absorbance spectrum to a geometry of the DNA. And if we can make those scans quickly enough, then we can get an assignment of the geometry for each time we make a scan, and then those geometries of the DNA, if we have a lot of them, then they become like a video, and we can see how the structure of the DNA is changing. An example of how this could be super useful is with the transcription elongation complex, which you can see in Figure 7, where many processes, like those listed in Figure 8, have rates from seconds to minutes to hours. And for those processes, this experiment could tell you how the geometry is changing with time, and also the geometries that it went through as it was changing. And this hasn't been possible in any experiments so far. So to conclude, the use of Cy3 as probes is useful for studying structure and conformational fluctuations in biological systems, such as dimer-labeled DNA forks and junctions in the transcription elongation complex. And single photon phase tagging techniques can be used to capture structural changes occurring at the single molecule level. And this information could help to clarify bimolecular mechanisms in protein nucleic acid complexes.